Hi everyone, how are you today? Hope you're feeling good. My name is Abdullah. I'm third year medical student. Today, me and my colleagues in Medicine College of Mosul third stage are talking our presentation about bacteriophage. I hope you will like it. I will give you a brief introduction about the subject. If you have any question, please don't be shy. We'll answer your question with all pleasure. Thanks Dr. Ahmed and Dr. Omar for supervise. What are bacteriophages? Bacteriophages or phages are viruses that can infect or lyse bacteria, regarded as the natural enemies of bacteria and can be used to fighting bacterial infection. Now talk about history. In 1986, Ernest Hankey reported that something in the waters of Ganges and the Yamuna River in India had marked antibacterial action against cholera and could pass through very fine filter. In 1915, British bacteriologist Frederick Todd discovered a small agent that infected and killed bacteria. He believed the agent must be one of the following. First, a stage of life cycle of bacteria. Second, an enzyme produced by the bacteria themselves. Third, virus that grow and destroy the bacteria. In 1917, Felix Herel discovered the agent killing bacteria independently at the Pasteur Institute in France. He observed that culture of the dysentery bacteria disappeared with the addition of bacteriophage filtrate obtained from sewage. Latterly, this was shown to be the bacteriophages. This slide pictures for Frederick Todt, Felix Herel, Ernest Hanken. Now my colleague Ali will continue the presentation.
Hello everyone, I am Ali, third year medical student, and I will continue by talking about bacteriophage structure and sources of bacteriophage. Bacteriophage structure, it consists of number 1, head. Head, it consists of genetic material, either single-stranded, double-stranded, DNA or RNA, surrounded by a protein coat called capsid, which protect the viral genome. Number 2, neck or collar, short region that connect the tail to the head. It consists of many discs. Number 3, tail. The tail sheath is made up of contractile proteins, which help an injection of genetic material into the host cell. Number 4, base plate, to which tail fiber and tail pins are attached. Sources of bacteriophage: human and animal intestine, running water, soil, effluent outlet and sewage, and corpses. Now, my colleague Mariam will continue.
Hi, I am Mariam Ammar, third year medical student. I will talk about the classification of bacteriophages. The classification scheme includes 1 order, 13 families, and 34 genera, and the families are distinguished taking into account the type of nucleic acid and virion morphology. Mainly, the bacteriophage distributed into 6 categories, which are tailed phages, non-tailed phages, DNA phages, RNA phages, filamentous, and pleomorphic phages, and we will talk about each group separately. The first group is the tailed bacteriophage. It represents the most numerous and widely distributed group of bacterial viruses. Actually, at least 5,000 phages were investigated by electronic microscope. The type of nucleic acid is double-stranded DNA, and the tail is arranged as a spiral structure or composed of discs, and is often terminated by special elements that facilitate the absorption to the surface of bacterial cell. There are distinctions between tailed phages in protein capsid constitute DNA prion structure, host range, serological and physiological characteristics. This group differentiates into 3 families according to tail characteristics, which are myoviridae, cyphoviridae, and podoviridae. Myoviridae, it includes 1250 phages with long contracting tail, as Enterobacteriophage D4. The second one, cyphoviridae. This family includes around 3000 phages with long non-contractile tail, as Enterobacteriophage lambda and Enterobacteriophage D5. And the last family, Podoviridae, includes around 700 phages with short rigid tail, as Enterobacteriophage T7. We can see in this picture the three families of the tailed bacteriophages. There are other criteria discriminating tailed bacteriophages at the genus level, such as the presence of DNA or RNA polymerase, atypical nucleotide, and nucleotide sequence. So far, 40 genera includes around 750 species classified on the basis of DNA hybridization, morphological trait, nucleotide sequence, and serological properties. Now my colleague Fatma will continue.
Hi, my name Fatma Saad, third year medical student. I will continue the classification of the bacteriophages and I will talk about non-tailed bacteriophages, DNA containing bacteriophages with polyhedral capsid, and RNA containing bacteriophages. Non-tailed bacteriophages are arranged into 10 families, often represented by single genus and species, morphologically sorted into three types: phages having polyhedral capsids, filamentous phages showing spiral symmetry, lastly, phages with capsids of diverse shapes and no visible axis of symmetry. The low density and sensitivity to chloroform and ether resulting from lipid envelopes or intracapsid lipid vesicles in many viruses. In this picture, this is the tailless phage which is surrounded by the capsid. DNA containing bacteriophages with polyhedral capsid composed of three families: Microviridae family, Tigviridae family, and Corticoviridae family. الممبر of الميكروفيريدي family infected the broad spectrum of bacteria, including the chlamydia, the spiroplasma, and enterobacteria, and it contains single-strand DNA. In this picture, this is the Tigviridae family, which display a broad host specificity range, including E. coli, Pseudomonas aeruginosa, and Salmonella enteric. In the last picture, this is the Corticoviridae family, which is characterized by intracapsid lipoprotein vesicle encircling double-strand DNA, infect gram-negative pseudo-Altiromonas strain. Lastly, the RNA containing bacteriophages with polyhedral capsid, the family of it are Leviviridae family and Cystoviridae. Leviviridae family, distinctive feature of it is the presence of single-strand RNA and virus specificity to chloroform bacteria harboring plasmid encoding F. pili, serving as phage of adsorption receptors. Viral RNA function as mRNA immediately after injection. Cystoviridae family, the virus are distinguished by the presence of an RNA polymerase complex and three molecules of double-strand RNA. The viron in the form of an icosahedron is surrounded by envelope and display specificity to phytopathogenic Pseudomonas. In this picture, this is the polyhedral virus, which composed of capsomeres and nucleic acid, and in picture B, this is the filamentous phage. My colleague Wasan will continue.
Hi, my name is Wasan Ahmed. I am third year medical student. I will talk about the rest of the classification, and I will start with filamentous bacteriophage. The family of this phage include Inoviridae, Lipotrixviridae, and Rudiviridae. The family Inoviridae based on single-strand DNA, and it's subdivided into two genera, genus Inovirus and genus Plectrovirus. The genus Inovirus consists of 29 phage species, morphologically represented by long flexible or stiff filaments, and their length determined by the size of the genome. The phage capable to infect Enterobacteria, Clostridia, bacteria of genus Thermus, and so on. The virons are sensitive to the chloroform and ultrasonic treatment, but they show a resistance to the temperature. The genus Plectrovirus consists of 15 species. Virons are short, straight rods and capable to infect only the mycoplasma. The last one, pleomorphic bacteriophage. The family of this phage include Plasmaviridae and Fusiloviridae. The Plasmaviridae, currently this family include a sole member, Mycoplasma virus type 2, infecting bacteria of genus Acoloplasma. As we can see in this picture, the virus lacking to the clear-cut capsid and consists of a solid nucleoprotide core surrounded by membrane, and the type of the nucleic acid is double-strand DNA. Fusiloviridae, the viruses of this family are characterized by the tapered capsid, ending up in a short spikes. Outer envelope is made up by two hydrophobic protein and lipids of the host cells. And the type of the nucleic acid is double-strand DNA. The most common organism is Sulfolopus shibiti SSV1 virus. To date, attempt to choose a suitable host for the propagation of the phage has failed. Yet it was found that the prophage might be activated by the ultraviolet or mitosine C. My colleague Faisal will continue the presentation, and thank you.
I'm Faisal Mohammed, third year medical student, and I will talk about the life cycle of bacteriophage, which is of two types. Firstly, virulent or lytic cycle. Secondly, we have the temperate or lysogenic cycle. The lytic cycle include firstly adsorption, secondly penetration, then we have the biosynthesis of phage component, after that assembly and maturation, finally release of progeny. Now we take stages of lytic cycle in some details. Firstly, we have the attachment of bacteriophage to bacterial cell wall, and that happens by specific receptor on its cell wall. Secondly, the bacteriophage penetrate the bacterial cell wall to inject its nucleic acid using its tail by contracting it, squeezing the nucleic material inside the bacteria. Then it follows that the synthesis of phage component. When the phage DNA get inside the bacteria, in this stage, the bacterial DNA degrades and phage nucleic acid transcription and protein synthesis get started. After that, we have the assembly to make new phages, which include the assembly of the head, in which the DNA incorporated inside it, then the tails and tail fiber. All these come together to make the new phage. Finally, the proliferating phase inside the bacteria lyses the bacterial cell wall and gets released in the medium. This slide demonstrates the lytic cycle in a brief. Firstly, we have the adsorption or attachment of bacteriophage to the receptors on the bacterial cell wall. Then the entry or penetration, DNA injection, synthesis of viral component, assembly and maturation, then the phage release in the medium. And now my colleague Dhilal will continue with you.
Hi everyone, my name is Dhilal Fawaz. I am third year medical student. I will talk about lysogenic cycle of bacteriophage. In this cycle, the bacteriophage incorporated their genetic material into chromosome of the host cell, resulting in prophage, and replicated as a single unit without destroying the cell. Under certain conditions, lysogenic phages can be induced to follow an lytic cycle. Now, this diagram will show the steps of the cycle. Here we can see how the phage attached to the cell wall of the host cell and injected their genetic material into the cell. Then the phage DNA incorporated into the host genome. After that, the cell organelles will be replicated, followed by division of the cell body, resulting to daughter cell, and the prophage DNA is passed on to daughter cell during every division. If a bacterium that contains prophage is exposed to stressful conditions like ultraviolet light, low nutrient or chemical substance like mitomycin C, the prophage DNA is excised from the bacterial chromosomes and enter the lytic cycle. At the end of the explanation of the life cycle of bacteriophage, I will mention the comparison between lytic and lysogenic cycle. In lytic cycle, virulent viral infection takes place. In lysogenic cycle, temperate viral replication takes place. In lytic cycle, produce progeny phages, while in lysogenic cycle, not to produce progeny. Bacteriophage DNA replication occurs independently in lytic cycle, while bacteriophage replication occurs with the bacterial DNA replication machinery in lysogenic cycle. Lastly, lytic cycle viral DNA will destroy the cell, while in lysogenic cycle not destroys the cell. And this short video explains the comparison between these cycles. Now my colleague Shahad will continue.
Welcome everyone, my name is Shahad Ziad, a third year medical student, and I will talk about the phage therapy. So what is phage therapy? As we know, phages are viruses that can infect and kill bacteria, so we are going to use them in the treatment of bacterial infection. So phage therapy involves the use of phages or their products as a bioagents for the treatment or prophylaxis of bacterial infectious disease, such as resistant bacterial infections. The efficacy and potential of phage therapy against multi-drug resistance. In this scheme, we are going to see the development of phage therapy according to years. In the box below, in 1915, they hypothesized phage as a virus, phage as a virus. In 1917, the discovery and characterization of viral nature of phages. In 1919, it was the first successful trial of phage therapy on dysentery patient, and from this, the concept of phage therapy started. And year after year, they initiated International Phage Institute in Georgia, then they use it in surgery in Poland, and use it in the treatment of cholera patients in India as intravenous. In 1940, they visualized phage using electron microscope, and in the same year in France and in the USA, they commercialization a phage therapeutic preparation. Till 1990, they demonstrated the role of phage in microbial turnover. In 21, they demonstrated the lysis activity of phage in vivo, and in 26, the FDA approved to use phage cocktail in ready-to-eat meat, and in 2013, they initiated Phagoburn project, which is a clinical trial of phage therapy, and in 2014, the European Parliament proposed phage as a complement to antibiotic. The requirement of phage therapy and the mode of administration. Phages use it. We are going to use the tailed bacteriophage. We have more than one type. We are going to use the tailed type in the treatment. We are going to use the lytic phages instead of lysogenic, and we are going to use host-specific phages because each phage attacks specific strain of bacteria. The mode of phage administration, we have more than one mode of administration. We can use these phages as intrarectally. We can use them parenterally as intramuscular or intravenous. We can administrate them orally or as direct release in water, and also as immersion or bath treatment. Immersion or bath treatment maybe if we have burn ulceration, and it is remain the mode of choice. Two approaches to bacteriophage therapy. We have two methods. The first is the intact phage therapy. In this method, we are going to use whole phage product, and we are going to use it to treat infections in patients with antibiotic resistance bacteria. The advantage of this method is the amplification factor. That is mean one phage will enter to the bacteria, replicate, releasing 10 to 100 particles, and then the bacteria will lysis, the phage will enter another bacteria, and so on. In this figure, we are going to see the phage infect the bacteria and replicate inside it. Finally, the bacteria will burst and the phage will attack another one. The other method is the phage components therapy. In this method, we are going to use part of the phage, which is the enzyme. Phage components that target bacterium may provide a boon to the anti-infective research. The most promising phage components is a class of the cell wall hydrolysis termed lysine. Lysines are purified phage-encoded peptide glycan hydrolysis that exhibit immediate, strong, and bacteriolytic activity when applied exogenously. In the figure, this is the bacteria. The small particles are the lysine. We will add the lysine to the bacteria. The bacteria finally will lysis and die. The benefit of phage therapy. The first and most important is lack of cross resistance with antibiotic. That is mean even if the bacteria resistance to antibiotic, phage can kill them because the mechanism of killing different from phage and antibiotic. They are the second is they are bactericidal agent. That is mean they will kill the bacteria. One of the benefits are few doses are needed because the amplification factor. They are also called auto-dosing, so few doses are needed. These phage are not toxic, they are safe to use. There may be an immune response. We can overcome this by using highly purified phage. And they are also highly specific. That is mean phage only will attack the pathogenic bacteria. The other bacteria, the bacteria of normal flora, won't be affected. So there is no chance to have another opportunistic infection like in case of using antibiotic. The other benefit is formulation and application versatility. That is mean phage can be combined with certain antibiotic. And they are also versatile in application form. We can use them as a liquid or a solid or creams and in more than one route of administration. Another benefit is to clear the biofilm. The biofilm, which is shield-like material released by the bacteria and make it resistant, so the phage can clear this biofilm and resolve the infection, and they are also less expensive. Now my colleague Munassam will continue, and thank you.
Hi everyone, I'm Munassam Jassam, third stage medical student. I will continue talking some examples about applications of bacteriophage. As we know, phage therapy involves the targeted application of bacteriophage-specific pathogenic bacteria. Due to rapid rise of multi-drug resistance, bacteria has led to renewed interest in phage therapy as a possible alternative to antibiotics, or at least as a supplementary approach to treatment of some bacterial infections. Here we describe the result of bacteriophage in certain examples. First of all, in agriculture, fisheries, and food industry, broad-range lytic phage Salmonella typhimurium 94 has been used for biological control of presence of Salmonella in food. Secondly, in medical applications, such as treatment of infections, like wound infections and many other infections. In treatment of wound infections, application of phage for 5 to 10 days provided positive clinical results in majority of wound infections treated by phages. The best results were achieved in treatment of infections caused by Staphylococcus species and Streptococcus species. Here we see in the picture diabetic patient with infected foot and potential development of gangrene. So we swabbing the affected tissue to isolate the pathogenic bacterium. Then we select the bacteriophage that colonize the pathogenic bacterium. Then we apply the phage to wound to treat it. Secondly, in treatment of infected burn, phage treatment is helpful in burn for treatment of infection due to biofilm, which is often resistant to multiple antibiotics. In the treatment of infected chronic ulcers, application of phage for up to several weeks provided positive clinical effect. It's also used to inhibit dental caries. Acid-forming bacteria in the mouth can be controlled by the use of bacteriophages. Streptococcus sanguis is the first colonizer of newly cleaned teeth, and other bacteria then attach to it. It forms 19% of teeth plaque. The destruction of Streptococcus sanguis by introduction of its parasitic bacteriophage will remove plaque from teeth surface. And many other infections can be treated by bacteriophage, such as bacillary dysentery, infections of skin, nasal mucosal infections, lung and pleural infections, peritonitis, osteomyelitis, and post-surgical wound infection, rhinitis, pharyngitis, inflammatory urological disease, and treatment of leishmania infection. Finally, bacteriophage is also used in treatment of cancers and gene delivery. It has been shown that bacteriophage can be harnessed for their potential in gene delivery to eukaryotic cell through transfection of toxic gene to cancer cell. Such gene delivery, along with chemotherapeutic agent, have great perspective in near the future. Phage display is useful in cancer treatment because it's easy to modify the phage by gene insertion to express desired anti-cancer protein or peptide on its capsid, showing anti-cancer activity or induce immune response. Various homing peptides have been reported against cancer-specific cells, tissue, and organs. The M13 and T7 phages were used for tumor cell targeting through tumor homing peptide identifications, such as RGD4C peptide, on the cancer cell and tissue-specific endothelial cells and prostate. It was also demonstrated that anti-cancer drug efficiency against breast cancer was increased when homing peptides were used with integrins for drug delivery without much toxic effect. Identification of tumor homing peptides and organ homing peptides can be helpful for targeting cancer cell, antigen detection, and drug delivery using nanoparticles. This approach facilitates the specific targeting of cancer cells, which can be optimized for targeted treatment or drug delivery through the bacteriophages. So what is the gene therapy? Gene therapy is a technique in which functional gene is delivered or introduced into the target cell to restore, overexpress, or inhibit desired gene product, which is now used to treat different cancers. Example, peroxidase, whose overexpression can lead to the death of cancer cell. The chimeric phage vector, named adeno-associated virus phage, is a promising candidate in targeted gene therapy. It is the hybrid of eukaryotic adeno-associated virus and the filamentous M13 bacteriophage, both having single-stranded DNA. It expresses 3 to 5 copies of the cyclic RGD4C ligand that can target alpha-5 beta-3 integrin receptors, mostly associated with tumor cells. So we can say that the use of bacteriophages exhibit enormous perspective in cancer detection, targeting, and imaging. Thank you so much, and now my colleague Safa will continue the presentation.
Hi, I am Safa, third year medical student, and I will talk about limitation of bacteriophage therapy. Not all phages are made for good therapeutics. Good therapeutic phage should have a high potential to reach and then kill bacteria in combination with a low potential to negatively modify the environments to which they are applied. So we have two types of phages, not temperate and temperate phages. Not temperate we define as obligatory lytic, which means it will release from the infected cell via lysis and unable to supply lysogeny, and this type is good therapeutics. While the temperate phages are bacteriophages that can choose between lytic and lysogenic pathway of development, and this type is therapeutically problematic. So why the use of temperate phages as therapeutics is problematic? This is due to a combination of display of superinfection immunity, which converts phage-sensitive bacteria into insensitive ones, and the encoding of bacterial virulence factors, including bacterial toxins. For example, Shiga toxin type 2 producing Escherichia coli causes significant disease. Treatment is supportive and antibiotics use is controversial. So if we give a patient an antibiotic fluoroquinolone, such as ciprofloxacin, this will lead to Shiga toxin type 2 encoding bacteriophage induction, and this will lead to increased Shiga toxin type 2 production from E. coli, which is the virulence factor, and this toxin causes people to have a bloody diarrhea, which considered adverse clinical consequences. Another phages that fail as therapeutics are phages that display poor killing potential against target bacteria due to either poor adsorption properties, low potential to evade bacterial defenses, or poor replication characteristics. Also, phages that display poor pharmacokinetics, that is poor absorption, distribution, and survival in situ. Now, what's the reason that phages are not unique pharmaceutical? That's because phages as pharmaceutical are protein-based live biological agents that can potentially interact with the body's immune system, can actively replicate, and can even evolve during manufacture or use, but are far from unique in these regards. Thank you, and now my colleague Karam will continue.
Hi, I am Karam. I am third stage medical student. I will continue talking about bacterial virulence properties altered by phages. We have to know that exotoxin production is the most widely recognized bacterial characteristic linked to bacteriophage infection. Phages can alter host bacterial properties relevant to all stages of infectious process, including bacterial adhesion, colonization, invasion, spread through human tissues, resistance to immune defense, exotoxin production, sensitivity to antibiotics, and transmissibility among humans. Now we talk about phages encode bacterial exotoxins. The most widely recognized example of phage-encoded virulence factor are exotoxins. Exotoxin production is the major pathogenic mechanism of several bacterial pathogens, including Vibrio cholerae, Clostridium diphtheria, and Clostridium botulinum. Note that phage-encoded exotoxin work by a variety of mechanisms. That means not all phage affecting all the bacteria in the same manner, but by a variety of mechanisms. Now we have to know about the role of bacteriophage in cholera and diphtheria. Firstly, in cholera. Bacteriophages that carrying the gene for cholera toxin infect Vibrio cholera, causing it to secrete cholera toxin, and this leads to severe diarrhea to occur caused by cholera toxin. In diphtheria, a phage that containing the gene for diphtheria toxin infect Clostridium diphtheriae, secrete diphtheria toxin, and this lead to inhibition of protein synthesis and cause diphtheria. This is the easiest way in explaining the role of bacteriophage in cholera and diphtheria. And finally, we have the conclusion about bacteriophage. The use of bacteriophage as a therapeutic agent can be considered as an old new approach for treatment of multi-drug resistant infections. Enormous number of phages provide enormous number of receptors on bacteria and other cells that can be targeted by phages. Selection of phage used in treatment should depend on the specificity of the phage biology in order to gain the required therapeutic effect and to avoid any unwanted harmful effect on body tissues. Gene delivery and phage display appear to be helpful in treatment of cancers. A huge body of research has been done on application of bacteriophage in medicine. However, more and more research work are required before being approved as a safe, effective medications in medical world. This is all about bacteriophage in a simple manner. And finally, thank you for all those who are listening to our seminars.